Okay, so this video is about how to choose the right cuvette for whatever it is that you're doing spectroscopically. Okay, so there are a couple of things to think about. The first is, are you doing absorbance or fluorescence? Okay, so absorbance or fluorescence. Absorbance, we're going to have a beam of light that hits a cuvette and continues to go on through. Okay, so it's going to pass right through the cuvette. For fluorescence, we're going to hit the cuvette, and instead of going on through, we're going to observe light at a 90-degree angle. Okay, so when you pick up a cuvette, um, these cuvettes tend to be square things that are either plastic or glass of some kind. And the first thing you want to notice is whether or not they're clear on all sides or whether they're foggy on some. Okay, so this is a plastic cuvette that is intended to be disposable, and you can see that it's kind of it's different on one side. That doesn't show up on the video too well than it is on the other. Okay, so with this particular cuvette, it's well, you guys should pick these up and take a look at them. If you run light kind of the way I'm holding it, then you get a 1 centimeter path. Okay, but to save volume, it's kind of squeezed down on the sides. Okay, so you don't want to go through that because then you don't have a regular path length. There are other cuvettes. Like I'll take this one out and show you. So this cuvette is going to be clear on all sides. Okay, so that's a fluorescence cuvette. It can be used for absorbance, but it doesn't have to be. Right? You can use it for fluorescence as well. We have some other cuvettes. So this one, can you see how that side is cloudy? I should tape down my background so that I don't move it around like that. That's going to be dizzying. If we flip it 90 degrees, that side is clear. Okay. So when you're doing the experiment, you want to set the clear side so that the light is going through the clear parts. Right? If you turn it around and you put the cloudy sides there, so if I now make that side cloud, see the cloudy side is in the path of the beam, then all you're going to see is the cuvette. You won't see any of your sample. Okay. So the first thing to think about is, are you doing absorbance or fluorescence? For absorbance, you only need 2 clear sides. For the fluorescence, you need 4. Right? Next thing to think about is, what kind of sample size do you have? Okay, so sample size. The cuvettes, I'll pass this one around. Go ahead and touch that by the cloudy sides. Okay, so that cuvette will hold 3 to 4 milliliters. Okay, so 3 to 4 milliliters for the full cuvettes. Okay, so this guy, it's a full cuvette. That one will hold 3 to 4 milliliters. This cuvette, which has a narrower bottom, will only hold, you can get by with half a milliliter or so. I'll go ahead and pass that around as well. Okay. The large ones are just referred to as cuvettes, but if you ever hear the phrase micro cuvette, then that's referring to something with a smaller volume. We actually have a couple of cuvettes that will handle samples all the way down to 50 microliters. Right? So depending on the volume of your sample, um, usually with these things, the having too much sample isn't a problem. Sometimes you don't have very much. Like if you're doing a protein purification or something, you might only have a couple hundred microliters of your sample. And then you don't want to use a whole lot of it to take a spectrum. Okay. Because even though you can recover it after doing a spectrum, sometimes you just don't want to. Okay. So second thing to think about is sample size. Third thing to think about is the wavelength range. Okay, so the wavelength range that you're wanting to use. And here you need to think about the material, right? So if we have glass, so glass is going to be transparent to visible light, but it might absorb some in the UV light. Okay. So typical optical glass is good from about 360 nanometers up to 1,000 nanometers. Okay. Then you can also have silica or quartz, right? And that does better in the UV. So that goes from 220, it's actually a much broader window, up to 2,500 nanometers. Okay. So if you're doing any UV work, then you want to be really careful about what glass you have. If you're interested in the visible range, like if your solution is colored, then just about anything works. Just about any cuvette will work. But if it's UV stuff, um, like you're wanting to quantify DNA at 260 or 280, or if you're wanting to consider a protein that absorbs at 280 nanometers, any of that kind of stuff, then you want to be really careful about what your cuvette's made of. Okay. And then there are all sorts of plastics, and just depending on the type of plastic you have, it may be UV transparent, it might not. Okay, so you want to just know what the cuvette is made of, right? Um, Now, in terms of handling cuvettes, you want to be careful to only touch the cloudy sides. Okay, don't touch the clear sides, because even though you can kind of clean up your fingerprints, you can't do that indefinitely. There is some damage that will be done to the cuvettes from that repeated cleaning process. Think about your glasses, if you have glasses. Like you can clean your glasses, but each time you do, you're rubbing stuff all over them, and it's just not a, you don't want to have to do that too much. It's better to just keep them clean than it is to make them dirty and then clean them again. Right? When you have a sample that is, or a cuvette that is clear on all sides, then you want to touch the edges, okay? And you want to touch the top and the bottom. So hold it like that, or hold it on the edges, not on the faces, okay? You want to be really gentle with these with these cuvettes. Um, Use gentle drying techniques. So I'm going to actually write that down. Write another note here, just so it's documented. Um, Gentle washing and drying. Okay. For instance, it's going to be tempting to you if you have some liquid in your cuvette. It's going to be super tempting to you. I'm getting a plastic cuvette to illustrate this. To turn the thing upside down and try to tap it out. Okay? It's also going to be super tempting to you to do that without actually tapping on a surface and maybe just kind of try and shake things out like this. That shows up really poorly on the video. Um, don't do that. Don't do that. Right? I have this is a cuvette that holds 50 microliters. So if you have just a really small sample, then this is the cuvette to use. And you can see that there's only a small little window where light can get through. I'm trying to orient it to the camera here. You can see it occasionally. Occasionally you can get light through there. But you guys, if I hold it up, you can see there's just a small window there for light to get through. We had, this is a new one, and the reason we have to get a new one is that over the summer, I thought it would be a good idea to try and get liquid out by shaking it. And it slipped out of my hands. So now we have half of a cuvette in the other lab. Just dingbat moves. Okay. So even if you're a careful old veteran of this kind of stuff, things happen. So you want to just be as gentle as you can with the cuvettes. All right. That's all I got on cuvettes.
How would you dry it?
How would I dry it? Well, so when you were in organic chemistry, how did you dry things?
With air.
With air. Okay. So air is, if you don't get too aggressive with the air, that can work. Acetone does a great job. All right. I would be careful with the acetone if you're using a plastic cuvette. Of course, using a plastic cuvette, like there's 100 of them in there for 20 bucks or something. You know, so if you're cleaning a plastic cuvette, I would encourage you just to stop and throw the cuvette away and grab another one. You know, don't do that.
Yep.
Don't clean. Life's too short to clean plastic cuvettes. Right? Just throw them away. The glass ones, though, like that cuvette that I just showed you, the small volume one, that's around $400. So, yeah, I felt bad when I broke it. You know, but we had that for. I'd been using that one for 12 years. You know, so it had a good long life, but it just did not survive the summer. So I would dry them by mixing in acetone, and then the acetone can do a really good job at getting the excess water out. If you are cleaning them, a little soap and like a Q-tip right down into the top of the cuvette works really well. Okay. You do not want to dry them with paper towels. Kim wipes work okay, but they're not great. But they're pretty good. Q-tips work okay for drying things, but definitely not paper towels. Paper towels will scratch. So when you're trying to think about cuvettes, think about what your technique is, what your sample size is, what wavelength you have, all of that stuff. And then you want to make sure that you're orienting it correct in your instrument. I think I'm going to go ahead and stop.